Hey Beaklings, I'm just going to go over glycolysis and the Krebs cycle here. This should not be where you're learning the material for the first time. Hopefully you already watched my video about drawing it out because I explained how the processes work there. And hopefully you already read these parts, these sections in your chapter as well. But once you feel like you have your head wrapped around glycolysis and the Krebs cycle, then you should continue and we can look deeper. Okay, so here we go. Glycolysis harvests chemical energy by oxidizing glucose to pyruvate. So you remember what oxidizing means, right? Take the electrons. So glycolysis means splitting of sugar. Glycolysis, cutting sugar. And one molecule of glucose is going to be oxidized to 2 molecules of pyruvate, right? So in other words, we're going to take the electrons here from glucose, thanks dehydrogenase, put them on the NAD+, thus reducing NAD+ to form NADH, and we're going to be left with 2 molecules of pyruvate. Okay, so this is actually not a terrible picture. Okay, I know it's a lot simpler than the one that we drew together, but once you understand the whole process of glycolysis, this one right here just shows you the main points, right? So you got 2 NADH, plus you have some protons released. You got 2 molecules of ATP, but mainly you're left with that pyruvate, and most of the energy of glucose is still right here, right? And don't forget, just like we did, they're only showing the C6 part of C6H12O6 right here for glucose. They're just showing the carbon skeleton, okay? And that's all that's going to be shown throughout here. But there's a lot more to it than that. Don't forget the oxygens and the hydrogens play a big role. All right. Now, how are you going to make that ATP? Remember, we talked about substrate level phosphorylation. So here you can see an enzyme fitting an intermediate in there. Okay, we'll talk more about those in a second. And you see that it has ADP also fitting in here to the active site. So all that's going to happen is this dehydrogenase enzyme, whichever one it is going to fit exactly this intermediate. It's going to remove the phosphorus from here, the phosphate group, and it's going to connect it to ADP, thus forming ATP and releasing the intermediate that's left. Right? Pretty cool. So where is NADH off to and why? Well, of course, the electron transport chain, right? As long as you have oxygen and you're doing normal cellular respiration, you're going to have these NADHs shuttled off to the mitochondria. Now, glycolysis is probably a very ancient process. It is pretty much ubiquitous, right? Basically everything does it, or just about everything. And it probably started not long after life began on our planet. So think about what life needs, right? You need some sort of heredity, right? So hereditary molecule out there, probably RNA when life first started. And you need a way to obtain and use energy, right? You need some way to do metabolism, right? Or metabolic reactions. So glycolysis, think about it. You don't need organelles. You don't need oxygen, right? All you kind of need is a little membrane enclosed area so you can perform these reactions to cytoplasm, right? Or cytosol. And we all have that in our cells, right? No matter if you're a prokaryote or a eukaryote. Okay, so very, very ancient process, and it must be pretty good because we're all still doing it today. Now, we did not draw glycolysis exactly like this. Okay, I shortened it up for you. And you're not responsible to know these titles of the intermediates over here. But what you should know is that there's 2 main phases to glycolysis. What I like to call the energy investment phase, or the preparatory phase, is when you lose energy, right? So we lost 2 ATP to start, or ATP phosphorylated glucose in this case, and ATP comes in again and phosphorylates an intermediate. Okay, so we're minus 2 ATP to start our chapter of trying to get energy. But that's okay, because now we're entering the energy payoff phase, right? So in the energy payoff phase, we get 2 molecules of NADH, and a better way to say that would be that, okay, we have G3P here, which again, you're not responsible for that intermediate name, but we're going to oxidize G3P, and by doing so, we're going to reduce NAD+ to form NADH. So NADH is in the reduced state. Okay, and not only did we get these high energy electrons in the energy payoff phase, but we also got 4 molecules of ATP out of there. Pretty sweet, right? So even though we spent 2 to start, we're already getting paid off more than what we put in. Now, having said that, of course, you had to start with glucose, which has a ton of energy in it, right? But every time you go from step to step to step, so if I get this back up for a second, right? These are all intermediates, right? We started with glucose, and intermediate, right? You just keep going through all these intermediates. So basically, the reactant of a step forms a product, right? That reactant is probably an intermediate, which forms a product, which is an intermediate, which then becomes a reactant, which becomes a product, intermediate again, right? So these intermediates are basically just step to step to step. You have to know, eventually you'll have to know all those molecules. But for us, no, we don't memorize that. Now, think about the 2 net ATP that you got. That's only about 5% of glucose's energy. But you also got some NADH, don't forget. So between that NADH, the 2 molecules of NADH, or the electrons in the NADH, right, and the 2 net ATP, you got 21% of glucose's energy out there in glycolysis. That's without any oxygen, right? So we have a lot more work to do to get the rest. Now, some people, well, I shouldn't say people, some organisms out there like yeast and bacteria, they're fine. They say, hey, I just got 21% of the energy of this sugar molecule. I'm good, right? But think about you. You have big brains, right? Some of us have bigger brains than others. And you have these big muscles and things like that. Well, sure, you need a lot more energy from that. You can't rely on just glycolysis to give you all the energy that you need, but some organisms can. Pretty cool. So let's see if you can answer this question then. So for each molecule of glucose processed in our cytoplasm during glycolysis, what are the main net molecular products? Again, 2 net ATP, right? 2 NADH, and 2 pyruvate. All right. So now we have our pyruvate, right? Well, we're not allowed to really do a whole lot with it yet, but at least we can get into the mitochondrion, right? All of what we've talked about so far happened in the cytoplasm. But now that you have pyruvate, you can get inside the mitochondrion, which you know is the powerhouse of the cell, right? As kids like to say. Well, the thing is, the pyruvate cannot get into the Krebs cycle or the citric acid cycle until you groom it, until you oxidize it. Okay, so what this is talking about is how do you get pyruvate ready to enter the citric acid cycle? All right, so there's 3 quick, easy steps here. First thing that has to happen is you need to remove a CO2. Okay, and because we're in this chapter, you know if you're going to go ahead and lyse off a carbon, okay, it'll leave as carbon dioxide, and dehydrogenase is sitting there saying, hey, hey, let me steal some of those hydrogens. Again, they're not pictured here, but don't forget there's lots of hydrogen left. And as you strip off the hydrogens, you take the electrons, and the hydrogens are left as protons. And you steal one of them right here, you carry it away, and the other one's just released. Now, what else has to happen? You're left then, if you lost a carbon, you only have 2 left, that's an acetyl group. Coenzyme A comes in here and says, aha, come here, acetyl group, I'll pick you up, and I'm going to take you to the Krebs cycle. So we did lose more energy. And one thing I haven't talked about yet this video is, oh boy, what didn't I mention yet? Heat. Right? Every time you're doing these metabolic reactions, every time you're taking electrons and transforming bonds and all this stuff, so much of the second law of thermodynamics comes into play because you're losing heat like crazy. There's a high level of entropy as you're doing these reactions. Okay, so even though we talked about the glucose's total energy before, it's not like you could ever get 100% of the potential energy of glucose to be used for you because no matter when you're doing these reactions, you're going to lose heat energy, right? And we can't use that. But now that we have acetyl coenzyme A, we are ready to head to the main oxidation furnace of your cells, which is the citric acid cycle. Okay, so the citric acid cycle completes the oxidation of organic fuels. By the end of this, okay, you saw as we drew our pictures together, you're not going to have anything left of your organic fuel, right? And of course, I keep talking about glucose here. It doesn't have to be glucose. There's this thing called beta oxidation. You can do lots of different things. There's lots of different sugars you can use. But point is, we mainly map out glucose so you can learn the whole processes. But there's other foods, of course, that you can break down as well. And a lot of times those foods that you break down will become intermediates for different parts of glycolysis or the citric acid cycle. But here we go. So coenzyme A basically helps bring this acetyl group in here to a guy named oxaloacetate. So he's a 4-carbon compound. And I don't want you memorizing all the intermediates just like in glycolysis, but I do think there's 3 you should know here. I think you should know oxaloacetate because he's kind of the end of the cycle and the beginning of the cycle. I think you should know acetyl coenzyme A, right? Because this acetyl group has to get delivered by the coenzyme A. And I think you should know that this 4 plus 2 equals this 6-carbon compound called citric acid or citrate. Okay, those are the 3 that I really feel like you should know. And now that you have citrate, we can go ahead and oxidize it. All right, so redox reactions now are going to go through this cycle, and they're just going to go ahead and steal the electrons, right? Dehydrogenase comes in. After the carbon dioxide is removed, you're going to strip the electrons here. You're going to go ahead and reduce NAD plus to form NADH, right? And of course you release a proton here as well. And then you're going to do it again, get some more electrons here. Actually, in this step, you make some GTP, which gets converted to ATP, which is pretty cool. So we have ATP being formed in the citric acid cycle, just a bit, right? And then over here, you have FADH2 being formed. So he's very similar to NADH, okay? Or in this case, FAD is like NAD+. But FAD gets reduced with these 2 electrons, and he actually steals 2 protons. He steals both H's in this case, okay, or hydrogen ions. And now in the last step, malate to oxaloacetate, we do it one more time. We're just rearranging bonds in this point, and you're going to go ahead and reduce NAD+ to form NADH. So this is a great furnace, right? We are burning our food to the point where the food that you ate is pretty much gone. By the time you're oxaloacetate, the food that you ate, well, where did it go? It's either carbon dioxide or water or whatever. It's basically the electrons and heat and some ATP. So, well, we need to get that energy out. We need more ATP, right? So let's take those electrons and do some good with them. Okay. All right. And yes, just like in glycolysis, don't forget this is substrate level phosphorylation, right? So you're going to fit in enzyme is going to help you make that, not oxidative phosphorylation at the end of your electron transport chain. So the Krebs cycle is complete when you rearrange malate back into oxaloacetate, and as long as the acetyl group is ready to come in again, thanks to coenzyme A, you're ready to go all over again, right? So this is why when you're working out and you feel your muscles getting nice and hot, a lot of you could think, oh man, my mitochondria inside the matrix must be doing a lot of work. Now, one other thing I'd like you to think about, if you remember when we drew our pictures, okay, which you should check the other video if you're forgetting that, is where is this happening? This citric acid cycle is right in the matrix, right? And it's probably right near the inner membrane. So the point is, here you are oxidizing the heck out of your molecules, and you're gaining these electrons. That NADH and FADH2, they only have to go a very short distance, right? Because the inner membrane is right there. They're right below the cristae, the folds of the cristae right now. Pretty cool, pretty neat setup. So what are our products here? Well, tricky. Some kids will look at the picture and say, oh, there we go, 1 ATP, 3 NADH, and 1 FADH2. But that's just one turn. Don't forget, we started with a molecule of glucose, so you should technically do this twice, because just like you had 2 pyruvate to get groomed before, now you have 2 acetyl-CoAs, right? So basically you get 2 ATP, 6 NADH, and 2 FADH2. All right. So comprende, can you do this? Let's see. How many net ATP, NADH, and FADH2 molecules were produced after a molecule of glucose was broken down in glycolysis and its products were processed in the citric acid cycle? Okay, let's break it down here. Let's start with ATP. How many ATP did we get from glycolysis was 2 net. Okay, so we got 2 net from glycolysis. How many did we get from the grooming? 0, right? A goose egg there. How many did we get from the Krebs cycle? Each turn was 1, so 2. So we actually got 4 ATP total, right? What about NADH? Let's think real hard. Well, in glycolysis, we got 2. In the grooming of pyruvate step, in the matrix, we got 2 more, so that's 4, right? And now in the citric acid cycle, 2 turns of that is 6, so that's 10 total, right? 10 NADH. And then how many FADH2? Well, that was just at that one step in the Krebs cycle, or the citric acid cycle, that we got 1, so 2 turns we'd get 2. Okay, so there you can see. I hope this video is a little helpful. Again, don't worry so much about memorizing little details, but I'll be honest, it does help. Okay, it does help to know every little step that's in there, but just at the level that we're going over. We're not going too crazy with the intermediates and things like that. But hopefully this helped, and hopefully you have a good understanding of glycolysis and the citric acid cycle. Take care.